Hi everyone, welcome to Chapter 5, Membrane Dynamics. A good chance we'll probably break this up into two pieces as well since I have 43 slides to go over and a couple of challenging concepts that we'll want to work on. To illustrate how important membranes can potentially be, this is a schematic of the coronavirus. And the coronavirus attacks certain cells just like any other virus does because of the characteristics of the membrane. The reason the coronavirus has a predisposition for lungs in many cases is due to the enzymes that are expressed in the lungs and around the lung cells. If this virus had a predisposition for liver cells, it might be a hepatitis virus. Or in the case of HIV virus tends to infect a certain member of our lymphocyte cells, the T4 helper cells, and that causes immune suppression. In some cases, it also will affect the neural systems, and so patients who are with advanced HIV often have neurological issues. So viruses have a predisposition for different cells based on the characteristics of the membranes of those cells, because the virus has to hook onto the cell and then pull itself and get its genetic material inserted into the cell. So that requires an interaction. And there are people that are actually immune to HIV because they lack the protein on their immune cells, and so the virus can't get inside the immune cells. We've actually cured HIV through bone marrow transplants from somebody who didn't have the protein into a person who had HIV. So about this chapter, we are going to talk about osmosis and tonicity, and this can be the tonicity side in particular we'll probably have to go over during live session. It's very difficult to animate, and I'm going to try to use a whiteboard to help illustrate the situation of tonicity. But it's very, very important because we want to give certain types of fluids to people who need fluid, but you just can't give any old fluid to everyone. If someone has hypertension or has a fluid challenge in their lungs and you want to deliver something intravenously, you want to give them a different kind of solution rather than just say a normal saline. So we're going to talk about that because it has very, very important clinical ramifications. We're going to talk about transport processes, in particular facilitated diffusion and active transport. At the end of this chapter, we're going to have sort of an integrated process of the sodium-potassium ATPase. Those transport processes include raw diffusion, but a lot of the time also include what we call facilitated diffusion, which is where proteins like channels and carriers that act like enzymes to allow things that normally wouldn't diffuse through a membrane to do so. Protein-mediated transport, that's what facilitated diffusion is, but also active transport is protein-mediated. Putting those together into a bigger picture, epithelial transport across an entire layer of one or more cells, transcellular transport, or perhaps paracellular. Vesicular, we're not going to talk about that much. So when you're reading it in your book, don't have to worry too much about vesicular transport. It's endocytosis, exocytosis, lots going on there, but it's really just, pinching off the membrane into a vesicle and then taking the vesicle and turning it back into membrane. Then finally, we're going to deal with resting membrane potential. So we're going to deal with these in probably part 1, and then we'll deal with these in part 2. Resting membrane potential is going to be saved for when we talk about neurons. So I'm not going to deal with it right away. We're going to save that section for when we get to neurons because that's where it really has important characteristics. So we're going to avoid vesicular transport and we're going to save resting membrane potential for later. So when it comes to talking about membranes, you have sort of two major areas that you are going to discuss. One is going to be biophysical factors, and the other is going to be mediated transport. I tend to start with the biophysical factors first, the raw characteristics of the membranes and how they can change a little bit if things change. And then we start adding proteins and showing you how things move. But again, you can start with one and then go back to the other. But we're going to start with biophysical factors. And before we go any further, before we get too deep, a couple of hard and fast rules for you. If you have a membrane, and this one doesn't seem to have any proteins in it, so we'll just take it for what it is. Generally speaking, hydrophilic molecules will not go through, but hydrophobic molecules will. So if we let those diffuse down their gradients, what we would see is that the hydrophilic gradient would never diminish, because hydrophilics or charged molecules like ions will not diffuse through a membrane unless it is allowed, permitted. Maybe we insert a protein or we change the characteristics of the membrane somehow to allow small ions to go through. But if it's hydrophobic, it will generally penetrate that lipid bilayer and move right through. I have an interesting story. I was looking for a free place to camp, and I was in Kentucky in the summertime. It was late at night, and I wasn't in the mood for finding a campground, and so I just found a field. I threw my tent out in the back of the field where nobody could see me, and in the morning I got up early so nobody would find me out there. And I'm walking through the field, which happens to be a tobacco field, and I just about got sick because nicotine was literally oozing out of these plants, and it's so hydrophobic it can penetrate skin, and that's why you can wear a nicotine patch on the outside of your skin. It's hydrophobic enough it can get to the bloodstream without any assistance, and I was suffering from a little bit of nicotine poisoning. On the other hand, let's say we have some molecule, and it doesn't go in because it's too hydrophilic or it's got an ion charge. If we somehow change that membrane, spread these phospholipid groups out, make it a little more watery for whatever reason, maybe that molecule can get through. That's the interface. In the previous model, if we put a protein in there and we could allow that ion through, or we can change the biophysical characteristics. But generally speaking, if it has an ionic charge, if it's aqueous, it is less likely to get through. And that's part of a larger thing called Fick's law. So selective permeability can be gain or loss as a function of the biophysical characteristic, what we would call a relative fluidity. At higher temperatures, relative fluidity tends to go up. Lower temperatures, relative fluidity tends to go down. And there are other ways that you can modulate it, but just to get that basic concept first. And again, hammering cholesterol here a little bit. Without cholesterol, if we have a lower temperature, it would mean the membrane is too rigid, can't allow things through that may be able to get through under normal fluid conditions, and may actually break and fracture, causing cell rupture. If it's too hot, the membrane is too fluid, it won't hold its shape, and now you have unregulated exchange of things across the membrane that normally couldn't. Regulating fluidity has life or death consequences, given that 2/3 of our total body water volume is inside our cells. Careful regulated exchange either between blood plasma and extracellular fluid and cells becomes really a critical component of physiology. And there's lots of numbers here. I'm not too worried about that. Let's add some pieces to the puzzle here. So intracellular fluid has high potassium, low sodium, low chloride. But it does have high proteins, and that contributes to the osmolarity inside the cell, running around 300. Interstitial fluid, which is also extracellular fluid, has relatively high sodium, high chloride, and I'm not going to worry about bicarbonate today, but low potassium, and we use energy to sustain that gradient across these two. Plasma, on the other hand, will have relatively high sodium and chloride, but also relatively high proteins. So get to know those, and as you think about osmolarity and so forth, you can consider those things. So even though they may be relatively similar in terms of their osmolarity, Nevertheless, these chambers, these compartments are in chemical disequilibrium. So sodium and chloride want to diffuse into the intracellular fluid. Potassium wants to diffuse out. Proteins want to diffuse out or out of the plasma in some cases. Every once in a while, if you have a problem with your capillaries, you'll start to see proteins spill into the extracellular fluid, and it'll begin to cause swelling in that compartment. So you could think about these things from that perspective and where this goes when it comes to inflammation or insufficiency of vascular function. But that's all extracellular fluid there in the red box, interstitial and plasma. So a reminder that homeostasis is a dynamic steady state. It is not an equilibrium. Now, by and large, most cells and compartments in the body are what we call at osmotic equilibrium. That is to say, they have the same osmolarity. If they didn't, water might leak across them and lead to swelling or loss of fluid in certain compartments. But by and large, most compartments are at osmotic equilibrium. A couple of big examples of where they're not are in the glomerulus of the kidney, but also in the colon. The colon, as we desiccate the remaining food material prior to excretion, we're at very much an osmotic disequilibrium, and the colon's very good at sequestering water. Most of the reason why we have osmotic equilibrium is that we have channels in the plasma membrane that allow water to rapidly equilibrate. Those channels are known as aquaporins. If we didn't have aquaporins, we would not see water move very rapidly across a membrane. It's the rapid equilibration, but because we permit water, it's facilitated diffusion. Not everybody will say that, but in our class, we will consider osmosis a form of facilitated diffusion by virtue of the aquaporins. We are very much in a chemical disequilibrium, and I showed you the gradients from the previous slide, so sodium and chloride, potassium, and sometimes protein. Also, those ions will contribute to an electrical disequilibrium, and that's going to be very, very important for signaling. The chemical disequilibrium may or may not result in an electrical one, but does allow for transport of material in many cases. Electrical disequilibrium is more about signaling and messaging. So osmosis is the movement, the diffusion, I should say, of water down its concentration gradient. So water will want to move to where solute is higher. So if solute is lower out here than in here, water will diffuse from an area of higher water concentration to an area of lower water concentration. And so that's osmosis, and we permit it to move through plasma membranes. We facilitate it using protein channels called aquaporins. And we can see osmosis at play. It is literally the interface between chemistry and physics and biology. It's a really, really cool phenomenon if you realize that you have water and it has chemical and physical properties, and it's going to affect biologic systems. So if we have like for instance, a dialysis membrane here, and we have different concentrations of solute, then what we will see is that water will have a higher concentration over here than here, and over time, water will begin to diffuse down its concentration gradient to the point that it will literally begin defying gravity. It is when gravity finally overcomes, that gravitational force is strong enough, it will counteract diffusion. Like I said, a chemical diffusional property that results in a physical pressure. If we put a wall here, like we built a wall right here, the water couldn't get any higher because literally the wall is pushing against the water, so it can't diffuse. So if we apply a force, that opposes osmosis, we can either slow osmosis down or stop it completely. So the interface between chemical behavior and physical pressure. So we call that osmotic pressure. It's the desire for water to move to an area of higher solute concentration, to an area of lower water concentration. So let's now figure out how to determine the link between osmolarity and tonicity. We're moving towards tonicity because that's the clinical aspect. So we have molarity, that is the number of molecules dissolved in a given volume of water. But not all molecules stay intact when they dissolve in water. Some may dissolve, but they stay intact, like for instance, glucose is an example of that. Glucose doesn't split into ions. But a lot of salts, what happens is they actually, when they dissolve, they dissociate. And so they will create more, every molecule in a salt may create more than one osmotically active, I say particle there, could be osmotically active entity, shall we say. So we can have a different molarity versus osmolarity. So for instance, sodium chloride, we're a common table salt, right? It has one molecule of NaCl, but when it is dissolved, it dissociates into a sodium cation and a chloride anion. And each one of these is osmotically active. So for every one molecule of sodium chloride, we have two osmotically active particles. So if the molarity of NaCl was 1, the osmolarity would be 2. Because the number of osmotic active particles is twice that. And we're going to deal with this in lab this week. So be prepared to do a little bit of riddles and twists on the difference between molarity and osmolarity. Because osmolarity is where tonicity begins to play. If you understand this relationship here, you can make the leap into tonicity. Occasionally, and I'm just putting this in as an aside, you'll see something saying osmolality. That's because in commercial preparation of fluids, it's easier to measure weight than it is to measure volume. They can measure it more precisely by weight. And the other thing is that osmolarity and osmolality only will begin to differ at relatively extreme temperatures and pressures. Here at near sea level or within the realm of human existence, most of the time those are the same. But if you're near boiling or something like that, osmolality can change. So it's particles per volume is osmolarity, like number of particles per liter. That's Avogadro's number. Osmolality would be particles of per kilogram of water, and under standard temperatures, a liter and a kilogram are synonymous. So I'm going to share with you a shortcut that I use, and that's the idea that that when molarity is expressed, that is the quote-unquote undissolved, or shall we say undissociated compound number of molecules, like the intact NaCl. So if we have 1 mole of NaCl in 1 liter, that would be 1 molar. But it would be 2 osmolar because it's like molarity, only it's the number of dissolved, dissociated particles in solution. So you take the molarity times the number of osmotically active particles that you know will form because of that when it dissolves, and that gives you osmolarity. 2 ions which are not osmotically active and don't normally contribute to osmolarity unless you're talking about really, really high concentrations. H plus and OH minus. So let's say we have NaCl and then we have an equal solution of NaOH. NaCl will be 2 times whatever the molarity is, NaOH will only be 1 because that OH interacts with the water molecules, and so it doesn't form an osmotically active compound. It actually interacts with protons and so forth and will form new water molecules. Same thing's true with H+. It'll form hydronium ions and become a part of the water, not dissolved in between, unless you're talking about exceptionally high concentrations, well beyond biologic relevance. So let's say we have HCl. HCl would have one osmotically active Cl and a non-osmotically active H. So the number here for the number of osmotically active particles would be 1 times whatever the molarity is. Same thing with NaOH. And so the book has some examples of comparing osmolarities. For instance, if you have 1 osmole of glucose, you have 2 osmoles of glucose, and 1 osmole of NaCl. Again, we've already factored in the dissolving. So 1 osmole is always going to be 1 osmole. So C is also 1 osmole, so they're isoosmotic or isosmotic. If you have more osmotically active particles, like solution B here, it's 2 osmoles, that would be hyperosmotic. More solute is more osmotic. Okay, more solute is less water. So if we have 1 osmole and 2 osmoles, this one's going to have a higher concentration of water than this one, and the water will want to diffuse if we allow it from the 1 to the 2. Got through the first part of this list here, and now let's move on to tonicity and osmolarity. They are related, but they have important differences. Osmolarity is the comparison of solute between 2 compartments. Tonicity is what's going to happen to the water, and that's a very, very important question because sometimes solute moves too. And if solute moves, that's going to affect tonicity. We might start with different osmolarities, but we're going to end up with a tonicity, which is the change in shape or size of a cell or a biological compartment because of influence by the fluid surrounding it. The key here is whether or not the solutes, we infer that water will move, if slowly, if at all, but it will move, and usually it's fast. The question is whether the solutes can make it through. If the solutes can make it through, we call those solutes penetrating. If they can't make it through, we call those solutes non-penetrating. And so osmolarity is a comparison of the particles in the solution surrounding the cells versus the inside of the cells. That's osmolarity. It's the concentration of water and solute outside the cells versus inside the cells. Tonicity in the biological context, now sometimes chemistry is going to differ on this, but in the biological context, tonicity says how the solution will affect the cell. So in a manner of speaking, you can say this is the before and this is the after. And again, it depends heavily on whether or not the solutes can penetrate or not. Now, the solutes inside the cell are almost always non-penetrating. You don't see solutes after you've gotten them in the cell moving back out of the cell. It's very, very rare. So we're going to assume anything inside the cell is non-penetrating. But what about the outside of the cell? If the outside of the cell has penetrating solute, that can make tonicity a little trickier than the straight up osmolarity comparison, if you will. So osmolarity is the chemical concentration, but the tonicity is how the cell is affected. So if we take some cells and we incubate it in a solution and the cells begin to swell, that means that solution A is hypotonic. Regardless of osmolarity, it's hypotonic. In solution B, the cell doesn't change size. Again, osmolarity doesn't matter here. If we don't see a change in size, it means that the solution is isotonic. And if the cell shrinks, again, regardless of osmolarity, the solution is hypertonic. probably one of the most difficult conceptual slides, at least in this first unit, if not in the semester. In this chart showing you how a hypoosmotic can only be hypotonic, but iso and hyperosmotic solutions can be more than just their equivalent. Like for instance, isotonic can be iso, but it could also be hypotonic. Same thing's true with hyperosmotic, though even more so. It could be hypertonic, it could be isotonic, or it could be hypotonic. Why the difference? Because some solutes are penetrating and some are not. When we talk about osmolarity, we're talking about the before the cells come in. If we have penetrating solutes, the osmolarity outside to inside is going to change. I don't want to worry about that change so much as the fact that if it has more osmolarity to begin with, then it's going to be hyperosmotic. Okay, there are some complexities here, and I'm going to try to dial it back a little bit. But if we have penetrating solute in our isoosmotic solution, it can then be hypotonic. And I'm going to try to use a whiteboard to help you illustrate it because it's really, really difficult to do in video like this. But I'm going to stop right there because we're going to move into diffusion and some other things in part B of this presentation. So thank you for your time. As always, if you have questions, reach out to me and I will talk to you soon. Thanks again.